Excuse me, sir.
Yes, yes, Arpit.
Sir, in the fourth question, can we make a table to differentiate that? Can
Which question? Question number
fourth, the differentiation.
4.
Yes, sir.
How is a primary oocyte different from a secondary oocyte? Yeah, you can just write it in bullets, like make a column, make a line in between 3 points or one, whatever points you want to read, 2 points on each side. Or you can also write it as one below the other. Anyway.
Okay, so that is what I was asking.
Yeah. Okay, people also keep a look at the time, yourself as well. I'm also keeping a look. So around 10-11 minutes are gone. Okay, we're left with around 20 minutes.
So the time will be over at 7:10.
Yes. Good evening, Alvia. Am I audible to you? You're not audible to me if you're speaking. Is Alvia audible to anyone? Yes. Alvia, am I audible to you?
Yeah, you are. Am I audible?
Yes, you're audible. So Alvia, we are having the test. You joined late, right? Why did you join late? Any issues?
Yeah, sorry, I didn't check my WhatsApp.
Okay, so quickly start with the test. Okay.
Okay.
It's for half an hour, but start it quickly. Okay, so everyone try to finish. Alvia, you can continue writing. Okay. Others, if you are done, then please, when you are sending it, you can send it as a PDF. That will be the best thing on the group because it will come as one single file. And please write your name on your sheet.
So I
Yeah,
sent it. Did you receive it?
Yes, I received Fatima's. So you need to write your name and your batch, which is MED and biology. Mention these three things, okay? So it becomes easier for me to segregate and check it. People, please wind up, everyone. Uh, apart from Alvia, you can take 5-10 more minutes, but not more than that. Okay. Others, please send your sheets in the group. I'm audible, right, everyone? Am I audible?
Yes.
Yes, please finish up and submit so that we can discuss and start with the proceedings of the class. Okay.
So what should I name the file as?
Just write the um, biology test. This is your second test for the batch, so you can write biology test 2 and just send it. Everyone, quick, please. It's already, I've given you 5 minutes above the time. So you need to submit now. Alvia, try to finish in next 5 minutes, okay? Okay, I've received Kulsum's assignment and Fatima's. When you are naming the file, also write your name in the file. Otherwise, it comes as some random number. Okay, everyone, quickly finish up now. Am I visible and audible?
Yes, sir.
Yeah, I have received Arpit's as well. Arpit, you have to send it on the group, okay? Please send one copy on the group also. Okay, Arpit, are you there?
Yes, sir.
Just forward it on the group as well.
Sir,
Send it on
nobody
the
sent it on the group.
huh?
Sir, I guess nobody sent it on the group, so I sent it on that.
No, nobody has, like, you are the first one. So send it.
Sir, did you get mine?
Not yet. Is it sent from your side?
Yes, sir. Which number was I supposed to send it to?
You have you are a part of the group, right? Where I send you
Yeah.
informations. Yeah, just send it on there.
All right.
I have not received Matthews or Arpit's. Okay, Alia and Alvi also. Please try to wind up and send it. Okay, cool, Alia. Okay, I received Matthews as well, the group. Alvia, how close are you to finish?
So I'm on question number 8.
Okay, just okay, do it. So everyone is probably thanking you at the moment, but please try to be on time, especially when there are tests, because you have to finish, but we lose time. Okay, do it. Alia. So I will start discussing in the next, let's say 4 minutes. Okay, Alia and Alvia. One, two. Okay, Alia, so do one thing. Just send on the group the pictures. I'll try to convert it into PDF. It's good if you can do this for me. It will save me some time. But if you're finding some issues with that, you just send the pictures. Okay. Take pictures and send it. So we have 1, 2. Alia, there are 4 pictures, right? I have received yours. Okay, it's fine. Like, just confirm that there are 4 pages, right? Okay. Alvia, so your time is over, Alvia. Like you joined, from the time you joined, it's already been half an hour. So please finish up and send it to me within next 1 minute so that we can start with the discussion. Okay. So those who finished in time or before time, like that's good thing. I hope you did not you did not leave any question. You should attempt all the questions in biology. And those who are taking more than the given time, they need to work on their writing skills. Alvia just joined late, so she's still in her time. But Alvia, your time is also over, right? It's half an hour. Please send.
So I'm set.
Okay, cool, perfect. The moment I'll receive Alvia's assignment, we'll quickly discuss and begin with the class. I think I received.
So can you discuss the answers first? I have to go in 15 minutes.
Yes, I actually want to discuss it fast because I also have to teach, right? We have to finish certain portions. Is this Alvia? You just sent your assignment as picture.
Okay.
Okay, this is still alias. Alvia, are you sending? Can we start discussing the
Yes.
answers? Okay, very good. So let's start with the discussion. So first question. Who found difficulty in which question? First question was state the role of epididymis in male fertility. Simple, right? What was the answer? What is the role of epididymis in male fertility? It is the site of functional maturity for the sperm, where the sperm matures functionally and gets the motility and viability factor. Correct? So you had to include these two terms: functional maturity, motility, and viability. If these three terms are in your answer, you will score full. Is it okay, everyone? Any doubts? Please stop me and ask if there is any doubt. I hope I'm audible and visible because no one is speaking. Yes, okay. Question second, what is the reason for the absence of menstrual cycle during pregnancy? Yes, Arpit. What is the reason? What have you written as the reason?
Yes, sir.
What is the reason? What have you written as the reason
So I have
for the
written that,
absence?
so the corpus luteum, it secretes large amount of progesterone and estrogen. So
Yeah, so the absence of menstrual cycle during pregnancy is that HCG is produced and lots of progesterone and estrogen is produced. So corpus luteum stays, and also the placenta starts forming progesterone and estrogen, which maintains the endometrium. That's right. Name the substance present in sperm acrosome and what is its function. So you can either name the specific enzyme, or you can, even if you have written hydrolytic enzymes, I'll give you marks. If someone has written hyaluronidase, I'll give them also the marks. And if someone has written lysine enzymes or lysins, that's also true. So the substance is hydrolytic enzymes. And what is its function? Function is
it enables sperm to break through zona pellucida.
Yes, perfect. To degrade the outer layers of the secondary oocyte or the ovum and to break into it. How is a primary oocyte different from a secondary oocyte? People?
sir,
What do you think? Yes.
The primary oocyte are formed when the female is still in the womb, and sir, the secondary oocytes are produced on the onset of puberty. So
Yes, but how is it different from each other? Like this is when you are saying when they are present in the female. Primary oocyte is present when the female is inside the womb, and secondary oocyte is present when the female is outside of the womb. Yes, Alvia. You're saying something?
Primary oocyte is surrounded by secondary follicles, and
Yes.
secondary oocyte is surrounded by tertiary follicles too.
Tertiary follicle or Graafian follicle, right? Yes, so that
And
is one difference.
The chromosome number of primary oocyte is 46, and for the secondary oocyte it's 23.
So for primary oocyte, yes. Remember I told you that primary spermatocyte or primary oocyte is diploid, and then it divides, it undergoes meiosis to become haploid secondary oocyte. So these were the two differences. If you are writing that one was present in the embryonic stage, one was present in the after birth, you are just telling where are they present. You are not telling the difference between the two. It is a difference between the host, the body, the mother. She was unborn and she is born. But you have to tell the difference between the two oocytes. Is it clear, Arpit?
Yes, sir.
Yes. What are the different stages of follicular phase of a menstrual cycle taking place in ovary and uterus? So what are the different stages of the follicular phase? So what are the different stages of the follicular phase of the menstrual cycle taking place in ovary and uterus? So basically you have to talk about phases. Follicular phase is specifically for the ovary. For uterus, you have to tell what are the, so there are three phases in each. The pre-ovulatory, ovulatory, and post-ovulatory. And ovary, which is also known as the follicular, ovulatory, and luteal phase. And in uterus, it is called the menstrual phase, the proliferative phase, and the secretory phase, right? So you just have to name those. No explanation was required. Explain in detail the various developmental stages of the zygote until implantation with suitable diagrams. Yeah, so there you could have started with a single cell, which is zygote, dividing into 2, 4, 8, 16 onwards. And from 8 to 16 onwards, it is called morula. So you have to mark that. And then you can finally show the the blastocyst stage where it implants, right? So in NCERT, this diagram is along with the diagram of a fallopian tube. If you make that, that is the best. If you do not make that, and even if only if you make the diagram for the developing zygote and embryo, that's also enough. Now, name the different types of cells present in the testes, differentiate between them on the basis of their function. Yes. So what are the different type of cells in the testes? One is Sertoli cell. Yes. The others are the basically developing spermatocytes. And outside of the seminiferous tubule are the Leydig cells. So you have to basically tell the difference between Sertoli, Leydig, and the spermatocytes. That's all. Explain spermatogenesis with the help of a flow chart. So I think this all of you could have done easily, just the flow chart. So starting from spermatogonia or primordial germ cell, if you want to do from there. Within the testes, it is from spermatogonia to the mature sperms. Describe the structure of a sperm with a diagram. What is the energy source of sperm cells? So in the diagram, you have to just label the structures. Even if you do not write about anything, it's okay, because it is the description with a diagram. So the diagram is self-explanatory if you label it correctly. What is the energy source of the sperm cells, people? This is easy.
Mitochondria.
Energy source
ATP.
is it's fructose.
Oh, yeah, fructose. Okay.
Okay. Energy source is from where you get energy. For every other cell, it is glucose, but for sperms, it is fructose, which is synthesized by seminal vesicles, right? That's why when this reserve of fructose is over, the sperms die after 48 hours. Yes, Alia is saying fructose, correct, Alia. How does the estrogen progesterone combination act as a contraceptive measure? Yes. Actually, this was a bonus question I gave you because you have already answered this question in question number 2. So the reason for the absence of menstrual cycle during pregnancy is estrogen, progesterone, high levels of that, because it maintains the endometrium. The same you have to write here. Estrogen, progesterone tricks the system, or it maintains the endometrium and do not allow further ovulation, because for ovulation to happen, the secretory phase has to come back to menstrual phase, and then proliferative phase has to begin. But it does not happen. So there is no cue for ovulation. So ovulation is delayed or it does not happen. Body feels like it is in a pregnant state already, but it is not, because the endometrium is maintained. So the same logic is same for question number 2 and question number 10. Is it clear, everyone? Fatima, you have any doubts?
No, sir.
So I hope all of you have attempted as best as you could, and you'll get the answer sheets within a day or two on the so yes, you'll get it within a day or two. I'll check and send it to you. Okay, so everyone, let's start with today's class. Any doubts, anything you want to ask before we begin today's class? Yes, no, something?
No, sir.
No doubts. Okay, so let's begin. Where were we in the last class?
We finished codominance.
Yes, we finished codominance. Can you all
So
see
I
this?
have to leave. Bye-bye.
Do you have an exam or something?
No, I have to go.
You have to go.
Yeah, I
Okay,
have to
so
go.
okay,
Bye-bye.
fine, Fatima. So get your hands on the video for this. Okay. Okay, everyone. So let's start with today's class. And I would like Arpit today. Arpit, are you there? Would you like to
Yes,
give
sir,
us
yes, sir, yes.
just a quick recap of what we did in the last class? And I think let's just start from the first law quickly. Just the law and the exceptions. I will be helping you if you get stuck.
Yes. So
Yep.
in the previous class, sir, we started with the law of dominance. Sir, the law of dominance, sir, it states that in heterozygous conditions, the allele or trait is expressed over another allele always. Sir, and the expressed one is called dominant, and the one which is not expressed is only recessive trait. So
Okay, good.
Then we did the that cross between tall plant and dwarf plant. So we did. Then you explained us about the pure line tall, hybrid tall, and dwarfs. Sorry. And then we did a question on heterozygous tall plant when it was crossed to the dwarf plant. Sir, then we started with test cross. So the test cross, it is a genetic cross that is done between a homozygous recessive and a dominant phenotype. Sir, whose genotype is to be determined, whether it is heterozygous or it is homozygous.
And you understood this, right?
Yes,
You had some doubts in this concept.
Yes, sir.
Okay, good.
Sir, then you told us about back cross. Sir, we told like it's the same process then backwards, like to back cross.
It's like going back to the parent generation and crossing it with any one of the parent. Simple, just back, one step back. Yeah. And then I told you that test cross is a special type of back cross where we choose the homozygous recessive parent. Yes?
Yes.
Yeah. Okay. Then coming to exceptions.
So then we did the exception. The first exception is the incomplete dominance. Sir, in this case,
what happens in this case? Incomplete dominance, right?
Yes.
It is not completely dominant. So what happens? Matthew, would you like to help your fellow batchmate here?
Yeah, why not? Okay, so incomplete dominance is an exception of the first Mendel's law of dominance. In incomplete an example of incomplete dominance is in the snapdragon plant in its flowers. So in the snapdragon plant, the homozygous dominant allele, which is a capital R, shows a red flower, and small r, a homozygous recessive, shows a white flower. But upon the breeding of these two flowers, you get a heterozygous capital R, small r, which causes pink flowers, which doesn't go with Mendel's law of dominance, which in this case should be a red flower, but we see that the alleles have blended to form pink.
Yes. Right. And hence the genotypic and the phenotypic ratio changes.
Yes.
And what is more affected in incomplete dominance?
The phenotypic ratio is more affected. Genotypic ratio stays the same.
Yes, right. And then coming to codominance.
Okay, yeah. So in codominance, both the alleles in a heterozygous condition is equally expressed, as seen in the blood groups of humans. In an AB blood group should actually show a dominant blood group either between A or B, but then both A and B antigens are present in AB blood group, unlike what Mendel's dominance rule says.
Yeah. Okay. Perfect. I can see Fatima is back. Fatima, you're back?
And yeah, sorry, sir. My class got cancelled, so I'm not going.
Okay, so you are with us. You're studying
I'm
genetics. Okay, very good. Okay, so in the last class we did till here. We thank you so much, Matthew and Arpit, for the recap. So all of you have understood codominance and incomplete dominance. Now, if you have done that, so this is one question that was asked in 2017 NEET. So it's a NEET question, and you have to just, it's based on the blood group, the blood type. Okay, so can you tell me the answer on the basis of codominance? Just solve this and tell me the answer right now. Can you read the question clearly, everyone? Let me read it for you. The genotypes of a husband and wife are IA, IB, and IA small I. So either you write IA and small I, it means the same thing as IA, IO. Okay, these are just two different notations. Is it clear, everyone? Yes. So
Yes,
the
sir.
question is asking, among the blood types of the children, how many different genotypes and phenotypes are possible? So please do it, make the cross, do it, and tell me which one will be the right answer. Whoever is done, just will send your answer in the chat. Just say option A, B, C, or D in the chat. The first one who will solve it will help me solving this, because this is an entrance question, so you have to be fast.
The 4 genotypes are there.
Is it Fatima?
4 genotypes and 3 phenotypes.
Okay, so I think Matthew and Fatima just answered the same time. Okay, so Fatima, you will help me. Okay, let's solve this. So for the husband, let's say for the male, it was IAIB, correct? Crossed with the female, who was IA, and for the sake of clarity, I'm writing IO. Okay. Then in the F1, we just have to make the Punnett square. Tell me what will be the different gametes?
IA,
or
and IB for the
Yes.
father, and then
Yes.
for the mother, IA and IO will be the gametes.
Yes. So what will be the first
IA,
possible? IA, which is
Then
blood group A, right?
yeah, and then IAI, and then
Oh.
IAIB,
Okay.
then IBI.
Yes, correct. So how many different types of, let's first tell about genotypic ratio, or how many different genotypes are present. You do not have to give the ratio here, but tell how many genotypes are there. One is this one. You can see that all the genotypes are different.
Yeah.
So if you have if a couple is AB, so this blood group is AB, and this blood group is A. So if a AB blood group marries a blood group, possibility of 4 different genotypes are there. So genotypes are 4. And how many phenotypes are there?
3 phenotypes.
A blood group. So these 2 will be A only, right?
Yeah.
A homozygous A and a heterozygous A. And then AB and B, right? So 3 phenotypes. Is it clear, everyone? Yes? Simple?
Yes.
Okay. Very good. So, okay, Ali is saying yes. Okay, very good. So codominance. So questions like this will come from the blood group. Questions like if the child is this blood group, what will be the blood group of the parents? What could be the possible blood group of the parents? These kind of questions can also come, which we will practice some of the questions later on. But let's move forward for now. So codominance and incomplete dominance are the exceptions of law of dominance, and this completes the first law. Now let's come to the second law given by Mendel, which is known as law of segregation. Okay, so in some books it's written law of segregation of gametes. This law is also known as law of purity of gametes. Okay, and writes few things about this law. This is the only law which is universally accepted, like it's universally true. So it's universal law or universally accepted law with no exceptions. Okay, why it has no exceptions? It has a reason for that. Let's first define what does law of segregation says. Write down. Law of segregation states, law of segregation states that when an organism makes gametes, each gamete receives a single copy of allele for each gene. A single copy of allele for each gene. Okay. In other words, you can also write, this law states that alleles are independently segregated. Alleles are independently segregated from each other to form gametes. Now, all this time when we're doing all the crosses, whether for incomplete dominance, for codominance, for law of dominance, we were actually following the law of segregation all the time. Do you see? Let's take this example. So this was a genotype, right? With 2 alleles. Do you see? So the father has 2 alleles for a given gene. Yes? And he received it from his father and his mother. Do you understand, everyone?
Yes, sir.
But while making gametes, it got segregated independent of each other. It never blends. Okay. Even in the case of incomplete dominance, let's look here. The presence of capital R and small r together shows blended phenotype, which is pink, right? But even when this genotype has to make gametes, capital R and small r get segregated independent of each other. Yes or no? So this shows blending, this also shows blending, but when gametes are formed, both the gametes are always pure. And when they again make combinations, see, this is the same R, right, which came from a blended parent. Yes or no?
Yes.
But when the same R, the same allele together formed a homozygous condition, they again give rise to red, but not pink. So which means that gametes never blend. Like at the level of gametes, there is no blending. It's always purity. So gametes are always pure. And that's a rule of biology. And that's why it has no exceptions. You understand, everyone? Is it clear?
Yes.
Law of segregation. Perfect. So this is all you have to study about law of segregation. Just know that it is pure law. Now coming to the third law, before coming to third law. Okay, let's first. Sorry?
Law of independent assortment.
Yes, Fatima, correct. It's called law of independent assortment. Now this law says the similar thing which law of segregation was saying about alleles. This is saying the similar thing about two genes. Okay, so law of independent assortment applies on two gene interaction or multigenic interaction. Okay, so to understand law of independent assortment, we first have to understand what is a two gene inheritance. So a two gene inheritance is studied through dihybrid cross, and a dihybrid cross is where name any two genes that you from pea plant.
Tallness and wrinkled seed.
Okay, tell me the character, height, and you're saying seed shape, right?
Yeah.
Okay, so let's say a pure tall plant with round seeds. Okay, so the plant is tall and has round seeds, is crossed with a dwarf plant having wrinkled seeds. Is it clear? Now we will do this together. So who will help me? Kulsum, help me please. Tell me the genotype of this parent 1. Kulsum, are you there?
Yes, sir.
Yes. Right, let's write the genotype. Tall plant with round seeds. What will be the genotype? It's pure line tall, pure line round. Okay, to begin with.
Capital T's.
Just one capital T?
Two capital T's.
Capital T, and for round seed? Capital R, right?
Okay.
Okay, so this will be the genotype for a two gene inheritance. If it was just one gene we were talking about, see, this is one gene with two alleles. Everyone understands, yes or no?
Sir.
So what will be the genotype of on this side, Matthew?
It will be small t, and smaller.
Smaller, and we are making a cross between them. Now the first, whenever we make a dihybrid cross, it's not like a monohybrid cross where we just are talking about one one gene, so that we can make simply we can separate the alleles and make the gametes. Here, to make the gametes, we have to look for all the permutation combinations. So the first step is to check for the genotype of the parent. What is the genotype? Then what different kind of gametes they can form. So this parent, how many different kind of gametes it can form? Any idea? So one allele from each gene will go into one gamete, right? One copy. So capital T will come for height and capital R can come for seed shape. Second option also is capital T and capital R only, right? There is no other option because both are homozygous for both the locus. Do you understand, everyone?
Yes.
So there'll be one type of gametes possible only. Here also it will be small t and small r. So when these two are crossed in the F1 generation, all the plant will be capital T, capital R, small r, right?
Yes.
And all the plants are hybrid tall and hybrid round, correct? Basically tall and round. No recessive trait is found here, just like following the law of dominance. Yes, this is F1 generation. But the interesting thing happens when you self cross F1. So let's self cross F1. Capital T, capital R, small r into capital T, capital R, small r. Correct? This will be the self cross. Now tell me, this is F1. Now tell me, what are the different gametes possible now?
Capital T, capital R, small t, small r.
So one will be this, other will be this, right? And anything else?
No.
Any other gamete possible, Fatima?
No, sir.
Yes or no?
No, sir. No. Small t,
It is
capital.
possible.
Yes, it is possible. There are, so these are the parental types. There are combinations possible, right?
Yes, sir. Small t,
Small t,
capital R,
yes.
what?
And
Capital T, small r.
yes. So there are 4 different types of gamete possible because there are this parent is heterozygous for both the genes. Okay, there are 4 possibilities. And for this parent also, the same possibilities will be there because they are basically, we are self-crossing, right? So now when you will make a Punnett square, it will not be just 4 progenies. On each side, now there are 4 gametes. You understand? So it will be a 4 into 4. Everyone understands why?
Yes, sir.
Because the possibilities increase now. So let's say for first parent, what are the all different gametes? Capital T, capital R, capital T, small r, small t, capital R, small t, small r, right? Same, capital T, capital R, capital T, small r, small t, capital R, small t, small r. Now tell me the offsprings. Kulsum, first one. Keep saying, I'll keep writing. Are you there, Kulsum?
Capital T, capital R square.
No squaring. It's not like
TR.
It's not. You cannot take common and cancel. That way you can take TR common, cancel, and in the bracket it's 1. No. We are using mathematics, but no, these are actual genes, okay? They do not get cancelled out. So it will be capital T, capital R, capital R. Okay. Same here, capital T, capital R, small r. Capital T, small t, capital R, capital R. Keep checking, people. If I'm making a mistake, please tell me. Capital R, small r. Capital T, capital R, small r. capital T, small r, small, sorry.
So capital
First we
T.
write, yeah, correct. Array. Capital T, small t, capital R, small r, capital T, small t, small r, capital T, small t, capital R, capital T, small t, capital R, small t, capital R, small t, capital R, small r. Just be careful when you are making the process. This is where you can make mistakes. Rest everything is easy. Am I correct?
Yes, sir.
Yes, everyone. Now, let's go for finding the genotypic and the phenotypic ratio. So I'll do it here. So first, what will be the genotypic ratio? Now, the genotypic ratio is going to be long here because there are many different type of genotypes present. Let's try to figure out. So this capital T, capital R is a unique genotype. There is no one else like this, right? No other genotype is like this. Yes?
Yes,
So
sir.
whenever you find genotype, I'm giving you a tip. Even in boards, keep a pencil with you and keep crossing the box that is done. So this is done. So this genotype is just 1. So I can write 1 is to. There is a way you go through diagonals. So this is a hint. I'm like, this is a cheat code. Just go through the diagonal and come through this. You will find the ratio easily. So this is 1. Then
Sir, how,
these
sir?
2, I'm telling you. So this is unique. Okay. The genotypes arrange themselves in a manner where on a diagonal they can be studied easily. This is just a trick. So the trick is, this is unique, right? This is 1. Okay. Then look at these 2. These 2 are unique, right? Like these 2 are same, not unique, but these 2 are same.
Yes, sir.
And there is no other genotype like this. So it is 1 is to 2 is to. Then this is unique. Capital T, smaller is to 1. Then these 2 are unique is to 2. Then this whole diagonal always is same. Can you see these 4 are same?
Yes, sir.
So is to 4. Then again we will start decreasing. These 2 are like same. This is unique. Then these 2 are same. And this is the recessive homozygous unique. So there is 1 homozygous dominant for both locations, 1 homozygous recessive for both locations, and in between are all the combinations possible. Do you understand? So the genotypic ratio becomes 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So this is the genotypic ratio in F2 for a dihybrid cross. What was the genotypic ratio for F2 in a monohybrid cross? 1 is to 2 is to 1, remember?
Yes, sir.
There also the same rule was being followed. It was diagonal, but because there were only 4 progenies, it was easy to see. But we start from 1, we go to the maximum, which can be 2, and then we come down to 1 because total number of offsprings are only 4. Here, total number of offsprings that are possible differently can be 16 different combinations because now there are 2 genes. So whenever we go from 1 gene to 2 gene, for 1 gene, the total number of offsprings were 4. For 2 genes, it becomes 4 raised to power 2. How many total number of offsprings? 16. Yes, people? Make sense?
Yes, sir.
If I am studying 3 genes at the same time, then 4 raised to power? How many total progenies then? If it is a tri-hybrid cross.
So what is a
64.
tri-hybrid cross?
This is a dihybrid cross. We are taking 2 genes to begin with. 2 genes, dihybrid. If I'm only crossing a tall with a short, it is a monohybrid cross, a single gene, mono. So we are hybridizing just 1 gene. Here we are hybridizing 2 genes together. So we get this one, a hybrid tall and a hybrid round. This is a 2 gene together. Trihybrid is where 3 genes are studied together. Okay. Tall, round, and yellow. If I make this cross, which capital T, capital T, capital R, capital R, capital Y, crossed with small t, small t, small r, small y, small y. This is a trihybrid cross. You understand? Now there are 3 genes to be hybridized.
We have a question like this.
In your exam?
Yeah.
Yeah. So we'll come to this after dihybrid. But you understood what is a trihybrid cross?
Yeah.
So in a dihybrid cross, the total number of combinations in F2 becomes 16 from 4. So in monohybrid cross, offsprings are 4 in F2. Okay. If it is dihybrid, 4 raised to power 2, 16. If it is trihybrid, 4 raised to power 3. So the F2 can have maximum combinations which are 64. You understand?
Got it, sir.
4 raised to power 3, 64, right?
Yes, sir.
No. Sixteen into 4 into 4 into 4. What is this? 64. Right. Select.
It's 64 only.
Yeah. Confused. So this is the genotypic ratio. What will be the phenotypic ratio, people? Now for phenotypic ratio, you have to figure out what could be the different possibilities. The different possibilities are the different type of gametes. You can take a hint from here. So either it could be tall round, yes, short wrinkled, yes, short round, and tall wrinkled. You understand? There can be 4 types of phenotype. You understand, everyone? Gametes give us
Yes.
the hint. So phenotype can be tall round. So I'll write, let's say, how many of them are tall round? Tell me quickly. How many are tall round? Let's keep crossing whatever is tall round. This is tall round?
1.
Yes. No, this is also tall round. 2. This is also tall and round. 3.
9.
This is also tall and round. 4. Yes, this is also tall and round. 5. This is not tall and round. This is tall and wrinkled. This is tall and round. So 1, 2, 3, 4, 5, 6. Then 7, 8, and 9. Right? So 9 are tall and round. Then next is tall and wrinkled. How many are tall and wrinkled? 1. This is tall and wrinkled. People.
Yes, sir.
Yes, sir.
This is tall and wrinkled. 2.
Yes, sir.
And this is tall and wrinkled. 3. So 9 is to 3. How many are? Next is
Dwarf and round.
dwarf and round. How many are dwarf and round?
2.
1,
4.
2,
3.
3.
3.
Only 3. So 9 is to 3 is to 3. And how many are dwarf and wrinkled? Just 1.
1.
So this is the phenotypic ratio. You understand? The genotypic and phenotypic ratio in F2.
Yes, sir.
I'm removing some of these green things so that it's more clear. Please note this down. And let me know when you're done. So you can use a pencil in boards and then delete, just like I did, clean. And you figured out all the ratios without.
So can we use a highlighter? Like
In boards, highlighter to highlight what? You
so
just
to
have to count
differentiate,
and then, but you just have to count, right? Highlighter will stay there, no? It is just for counting, just count and then remove. This 2 is number of genes. And this 4 is progenies in F2 in a monohybrid cross. You can also remember this. Is it done, everyone? Let me know when you're done. Okay, done. Fatima's done. Others? Done or still doing? Okay, math is done. Okay, so let's move forward. Shall we? Just from here, giving you a taste of a trihybrid cross. Let's see if this is the question that a trihybrid cross is being made. So it's clear that in the first, so if these are the parents, first cross is always easy. So from parents, when you cross a pure line parent, pure line dominant with a pure line recessive, how many number of genes they have does not matter. There will be only one gamete type possible. In this case, it will be T, R, and Y, capital T, capital R, capital Y, right? Which will be crossed with small t, small r, small y, correct? This is the only possibility because they are homozygous for all the locations. Yes or no?
yes, sir.
Yes.
So what will be the F1 here? Capital T, small t, capital R, small r, capital Y, small y. Yes, very good, Fatima. So this will be the F1. Now, when you self cross F1, now I'm going to tell you a shorthand trick. When you self cross F1, basically what are you trying to do? Capital T, small t, capital R, capital Y, small y, crossed with capital T, small t, capital R, capital Y, small y. Again, the first step is to find how many different gametes are possible. Can you tell me how many different gametes are possible? Let's first do it the tedious way, then I'll give you a short, like a trick. One is capital T, capital R, capital Y. Yes. The other is small t, small r, small y. Let's first write the parental types. This is going to be there, right? Now the parental. Capital T, capital R, but small y. Capital T, but small r and small y. Yes. Small t, capital R and capital Y. Small t, small r and capital Y. Others? And small t, capital R, small y. And can you think of any other combination?
Capital T and no.
Capital T? Yes, yes, you're right, you're right. Tell me.
Capital T, small r, capital Y.
Yes, right.
Yes.
And
Sir, I wanted to say this.
you were saying right thing, Arpit. I told you, come on, but and let me tell you, there is one more possibility. Now, whoever finds that will get appreciated by me. What is the other possibility? One more.
Capital T, capital R, small y.
Capital T, capital R, small y. It's here. Arpit, it's here already. Small t, capital R, small y. Yes.
Yes, sir.
Now, please confirm. Please
Sir,
confirm
it's
if
already
all.
there.
Where?
The third
Where?
last.
Third last.
Yeah, it's already there.
Yes, sir.
Yeah.
Same.
Yes, very good. So which one is missing? That's why we are confirming. So sometimes making gametes become very tedious.
Sir,
I
it's
also don't
difficult.
follow. I also don't follow this method. I'll just tell you the shorthand trick. You just need to know how many different types of gametes are formed for trihybrid onwards. Because if you go on making a cross, it's going to be long one. So they are heterozygous for 3 locations. Can any 1, 2, 3, 4, 5, 6, 7, 8. Anything else that is not all are capital, all are small. And so we are missing something. What are we missing?
So maybe if you do it from the start again, maybe you'll get it.
We do it from the start. I think we have made all right.
So it's not like genetic.
It's not?
I tried every permutation combination. But it's not.
So this is the only possibility, right?
Yes, sir.
Are you sure? Yeah, I think we did everything correct. Yes. Okay. So how many are they? 1, 2, 3, 4, 5, 6, 7, 8. Yes, that's correct. So if this is 8 on one side, then there has to be 8 on other side, right?
Yes, sir.
8 8s are
64.
yes, we will get 64 combinations. Yes, there'll be no other combination because through this formula, if it is a trihybrid, you just take it as 4 raised to power 3 and it becomes 64. So when you are making a Punnett square, there has to be a squaring, like, right?
Yes, sir.
So 8 and 8 will make 64, right?
Yes, sir.
If it is a tetra hybrid, let's say, there are 4, there are this. Now, can you quickly tell me how many different gametes are possible? Those who understood. how many gametes are possible for a tetra hybrid cross? Okay, I received one answer.
Sir, 256.
What? Those will be combinations. 256 gametes cannot be formed, no? Just by
I was talking about combinations.
I'm talking about gametes. How many gametes, different type of gametes will be formed? So first you will do 4 raised to power 4, which is
16.
no. 4 raised to power 4 is not 16. 4 into 4 is 16. 4 raised to power 4 is 64 into 4. What is 64 into 4?
256.
Right? So if you have total 256, so Arpit, you are saying 256 for the total combinations, right?
Yes,
so
sir.
if you have total combinations which are going to be 256, how many should be the number of gametes?
16.
Correct or no? There has to be 16 on each side.
Yes, sir. Yes, sir.
So that you get 256. So this will give 16 different type of gametes. Do you understand how to find this shorthand trick? No need to go into making all the permutation combinations. You understood, Fatima?
No, sir.
See, is it clear? Now you can do for any.
Understand.
You understood?
I
Or you
didn't.
did not? You did not understand. Okay, let's do it again then. Now, Matthew, will you please help me?
Sir, it's
Yes,
quite
sir.
difficult.
Yes, so let's start with trihybrid. You understood for dihybrid? Fatima, how many different gametes are possible in a dihybrid?
4.
4 maximum. On the other side also 4. So 4 fours are 4 into 4. Total combinations? 16 combinations, right?
Yes.
You understand, Fatima?
Yes, sir.
Now there is a formula. There you have to write 4 raised to power. If it is a monohybrid cross, you will write raised to power 1. So how many combinations if it is a monohybrid cross?
4.
So this is the formula to find number of combinations in F2 generation. So number of combination if it's a monohybrid cross is 4. If it is a dihybrid cross, the number of combinations are 4 raised to power 2.
16.
Yes. If it's a trihybrid cross, then the number of combinations will be Fatima. It's for you, the question. Because you did not understand, so we are doing it like this. Number of combinations are going to be
64.
64. If it is a tetra hybrid cross, then number of combinations are going to be 4 raised to power 4.
256.
256, right? Now, when it was 4 combinations inside the Punnett square, if there are 4 combinations, how many gametes were there on each side?
4 gametes.
2. 2 into 2 gametes on each side. It was a monohybrid cross. If there are 4 gametes, then it will be 16. If on one side there are 4 gametes, Fatima, then on the other side also there will be 4 gametes, and total number of combinations will be how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Sorry, 4, 8, 12, 16. So if it is 4 by 4 ka cross, the number of combinations will be 16. But if the number of combinations are 4, what will be the cross? 2 by 2, right?
Yeah.
If the number of combinations are 64, then it's 64 here. So what should be the cross? What by what? What number multiplied by the same number gives you 64? 8 8s are 64, right, Fatima?
Yes, sir.
So in a trihybrid cross, where it is 4 raised to power 3 for trihybrid cross, maximum 8 gametes are possible on each side, giving rise to 64 combinations. Yes? Is it clear?
Yes, sir.
Same way, if it is a tetrahybrid cross, we already found the number of combinations by this formula. If you found the number of combinations, which you only said is 256, so I'm asking you, if there are 256 combinations, which will take you like, let's say, half an hour to make. So if you go by just making all the combinations, you will make mistakes, not be able to find the right answer. But we know that there are 256 combinations possible. So what number multiplied by the same, because whatever will be here, the same gametes will be here. What will give you 256? What is the square root of 256? In short, I am asking. Can you find square roots? Of 256? What was the square root of 64? 8, right?
Yes, sir.
What is the square root of 256?
16.
16. So there will be 16 gametes on this side and 16 gametes on this side, and they will multiply to make 256 combinations. So in a tetrahybrid cross, how many gametes are possible?
16.
Now this is for all. If there is a pentahybrid cross, how many gametes are possible? Quick. So you have to do 4 raised to power 5 is equal to 4 raised to power 5 is equal to yes, Alia. 1024, correct. So just to find the number of gametes now, you have to find the square root of 1024. What is the square root of 1024? Quickly.
32.
32 into 32 will give you 1024 combinations, right? So how many gametes are possible if it's a pentahybrid cross? 32. You understand, everyone?
Yes, sir.
Pakka?
Yeah.
So this way you do not have to go on a hunt to make all the combinations and look which one is made, which one is repeated. Okay. So this is how we find it. Just remember this formula. I've written it here. You can write it down. It's to find number of combinations in F2. Once you find number of combinations, you can just do the square root and know the number of gametes. Perfect. So coming back to the third law of Mendel, which was, so you understood dihybrid cross, trihybrid cross, tetrahybrid cross. In NCERT, they will quiz you mostly till dihybrid cross for the ratios. Beyond dihybrid cross, if there is a question that comes, just remember that the question will not ask you the ratio. It will only ask you how many different types of gametes are possible. Okay. Is it clear?
Yes, sir.
Yes.
Okay. Another way to directly find the gametes, so in this case, look here carefully. This parent, which we self-crossed to find gametes, it is heterozygous for all the 3 locations. Yes? Yes or no?
Yes, sir.
If, let's say, let me give you some questions. So I'll explain you this. Let's say if it heterozygous only for 2 locations. Now, how many gametes are possible? If they are not pure line parents, you cannot go through the combination way, because clearly we are not asking about F2 now. In F2, we self-cross hybrid, F1 hybrids. Now, this is not a hybrid for all the genes. For one gene, it is homozygous. So how many gametes will be formed? For this also, I'll tell you a shorthand. But first tell me, one possibility is T will always be capital T. TRY, we are going through the same headache, but it's important to learn. Then T R small y. Then T will be T only. Small r small y. Then T. What else is possible?
Smaller, capital.
Smaller, capital Y. And anything else which is possible? Arpit will tell me. For this, how many gametes are possible? We have figured out 4 till now. Anything else which is possible? No, right? Arpit is gone, okay? Oh yes, Arpit is not here. Matthew.
Yes, sir.
Any other combination that is possible for gametes? No, right?
No, sir.
So how many gametes? 4 gametes. This is also a trihybrid parent, but it is homozygous for one gene and heterozygous only for 2 genes. So there is a formula to directly find gametes for any number of for any parent. It should not, like, not necessarily it has to be a pure hybrid parent, any parent. So write down this formula, it will be very handy. Formula to find number of gametes by any genotype. You have to count the number of hybrids, okay, hybrid genes. So how many hybrid genes are in this trihybrid parent?
Only one.
2. Why only one? Only one is homozygous.
Sorry, I thought you were asking homozygous.
No, hybrid. How many are heterozygous?
2.
2, right? So there are 2 alleles for every gene raised to power number of heterozygous genes or heterozygous alleles. So in this case, it is 2 raised to power 2, which is 4. You understand? Everyone, it also applies here. How many heterozygous alleles are here in this parent?
3.
So 2 possibilities for each gene. There can be 2 contrasting allele raised to power. What is 2 raised to power 3?
8.
2 into 2 into 2. It is
8.
8. Is it correct with what we did? 8, right? Yes.
Sir.
Tell me, now this is a question for all of you. With this, I will end the class. Tell me how many gametes will be formed by this parent. Total number of gametes. Two raised to power. How many are heterozygous? 1, 2,
4.
3, 4. So 2 raised to power 4, which is Quick.
16.
Yes. Total gametes will be 16. Though it is a 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7. It's a septa hybrid cross, but still there will be only 16 gametes because there are only 4 locations, 4 genes which are heterozygous. If all these 7s are heterozygous, you can do it raised to power 7, or you can go through the combination method that I told you. But this one is more.
If them are heterozygous, then we have to do the combination method, right?
If all are heterozygous.
No, if all are homozygous.
If all are homozygous, then there will be only one gamete.
Oh, okay.
If all are homozygous, which you are saying this, right? So there will be only one gamete, TYRSPQZ.
Yes, sir.
Yes, don't get confused. If all are heterozygous, then either you can use this as well to find directly gametes, or if you have to find the number of combinations, then you can do 4 raised to power. What is this? Hepta 7. And you can find total number of combinations. So see what they are asking in the question. If they're asking the total number of combinations possible, or if they're asking total number of gametes possible. So if all are heterozygous, then total number of combination can be found through this. And if there are some heterozygous, some homozygous, then you first found the total number of gametes, and to find the combinations, then multiply the gamete by itself.
This is
Or
such
square.
an easy method. If I knew this in the exam, I wouldn't lose 1 mark.
Okay, now you will not lose 1 mark. Your teacher didn't tell you in school this? I think this school teachers do tell. I think students don't. They didn't tell you? If not, then you go and tell them to tell this to the class. It's simple. It's very easy. It's for entrances, basically. Okay, everyone?
Yes,
So
sir.
we are beyond our time now. So with this, I declare the class here. So you go and practice more on these 2 formulas. 2 raised to power x, where x is the number of heterozygous alleles, or 4 raised to power x, where x is the number of genes. And to use this formula, all the genes that you are taking, it has to be a heterozygous for all the locations. It has to be a pure hybrid parent. Okay, to find. So this gives you combination